when I was a sophomore in college, my friends and I decided it would be a great idea to live all 7 of us in a 3-bedroom apartment. Yes, we were a little squished, but we were also able to support each other through the highs and the lows. During the first winter, one of the guys started to rapidly lose his vision. We soon found out this was the result of an autoimmune disease called multiple sclerosis. This experience got me really thinking about autoimmunity. For 19 years, my friend's immune system was functioning perfectly. So why did his immune system turn from his best friend into his worst enemy? This is where my research begins, but before we jump into that, let's just take a step back and think about what happens when an immune cell decides to target and kill another cell. I personally like to think about this like Jack from The Incredibles. He can quickly turn from a calm child into a dangerous weapon when he gets angry or scared. This is basically what happens in our immune cells. When they see something foreign, like a virus or a bacterial cell, they can dramatically change their behavior, turning from their resting state to an active state to fight it off. Now, the way that cells change their behavior is by turning on or off thousands of genes. So I specifically studied what regulates these changes in gene activity. Just like stoplights control traffic, there are proteins in our cells that regulate our genes. If there was no regulation, there would be complete chaos. Maybe this reminds you of driving in LA. Good regulation is critical, yes, for our sanity on the road, but also for proper immune cell activation. What's unique about my research is that instead of focusing in on one gene that has been correlated with disease progression, I study how every gene in your genome is affected and regulated during immune cell activation. This is like gazing out on the whole landscape instead of focusing in on a single blade of grass. This approach allows me to search for logic and patterns in my data. So far, I have identified unique roles for proteins that are key in regulating immune activity. This means that proteins that are currently thought of as unnecessary or redundant, I find actually have distinct roles in regulating your immune system. This research could uncover new and precise drug targets that could, in the case of autoimmune diseases like multiple sclerosis, restore the function of your immune system as your best friend. Thank you.
Amazing, right? Just most of you guys can understand what she's talking about, right? But we're doing like things that we don't understand even ourselves, to be honest. Yeah, but she did it so well, and it was so surprising for me, and it's a shock for me. And she's just about your age, and she did it so well, and so appealing. So she can pitch herself. So once when she get into her own business, like her future career, that will be part of her really powerful weapon, right? So that's the ability I think you guys should get it. Because no matter what kind of doctor or no matter what kind of career that you're going to pursue in your future, it's a necessary skill. Yeah, so at the moment I saw her presentation, I decided the next year, I have to do it. I have to do it. And it's a great determination because I know it will be super tough for me. I am not a native speaker. Okay, so this is how things go. This kind of presentation is limited to 3 minutes, only 3 minutes. And the slides should fewer than 3 slides. So you have to be clear, you have to be really concise, and you have to be appealing or attractive to the audience. Okay, and the participant could be like grad school from any kind of, like grad student from any kind of grad school. So it can be like business school, it could be music school, it can be anyone, any kind of graduate student among the whole campus. So you're competing not to the student among the biomedical field, but you're also competing to the student coming from like music school or science go out. We have a lot of different kind of interesting competitor. But most of the time, we biomedical students won the prize, most of the time. I don't know why, but it's most of the case. Okay, so this is our 2019 winner. She is the video that I show you, she won the second prize. And this is the first prize. So it was so amazing, like, okay, she is so great. And she was not the first prize. How good could be the first prize be, right? That would be really a great question. So let's take a look at her presentation. To be honest, I don't think she is as good as my colleague, but she's still doing a pretty good job.
But don't worry, I won't use my 3 minutes to talk about it. Whether you're buying Girl Scout cookies or donating to research hospitals like St. Jude, one thing's the same, and that's the desire to help kids. At the heart of my research is a strong desire to help kids with Duchenne muscular dystrophy, or DMD. DMD is the most common fatal muscular disorder primarily affecting boys. 300,000 boys are affected worldwide. In the US alone, 24,000 boys are affected. That's like half of the students at UCLA. DMD itself is caused by a change in DNA that results in progressive muscle weakness. These kids are typically diagnosed before the age of 5 and are wheelchair-bound by age 12. Since our diaphragm is also muscle, boys with DMD can face respiratory complications. And if left untreated, common infections like pneumonia can have dire consequences for these boys. And this has led to the heart problems that boys with DMD face. Now, heart failure is the #1 cause of death of boys with DMD. But all hope is not lost. In order to reduce this frightening statistic, this just means that we need to look at the heart a little bit closer in order to better understand how this disease affects the heart. And my research aims to do just that. And by look a little bit closer, I mean really close. We can do that using magnetic resonance imaging, or MRI. One advantage of MRI is that we can look at the heart from many angles, including the cross-section on the right. The dark circular region, that's the muscle that we're interested in looking at. Unfortunately, MRI is not routine in DMD. So, by the time that symptoms of heart dysfunction are recognized, it's often a little too late for MRI to be as useful as it could be. We also don't know exactly at what point in time the heart becomes affected in DMD. And that's the specific question that I'm looking to answer with my research. Looking to answer this question by analyzing hundreds of MRI images from boys with DMD and healthy volunteers at all ages. I've developed this analysis tool that utilizes color maps in order to look for patterns of muscle damage in the heart. And so far, I've seen that in a healthy heart, the muscle region appears more uniform in color. But for boys with DMD, you see an irregular color pattern, indicating where the heart is more affected. These results are really exciting because this tells me that MRI can pinpoint the time the heart becomes affected in DMD. Moreover, this is also exciting because doctors all over the world can use my analysis tool to order MRI exams and look at the disease progression of their patients over time. We can also use MRI to evaluate existing and new drug therapies aimed at helping boys with DMD. I've had a great time telling you all about how my research efforts are on track to helping kids. I hope I've inspired you to help kids in your own way today. Thank you.
So this is a pretty large event or very important event among the campus for most of the grad students. So after their inspiration, I told myself I should do it. Like I have to conquer the kind of stage fright that I got in my first class. So after 2 years, I decided to face what I'm really afraid of, talking or speaking English on the stage. So we have 3 rounds. The first round is preliminary rounds that we will get rid of half of the student. And for the second round or semi-final, we will get rid of the most of the student there. And we have only 10 finalists that will get into the final round. And what's also attracting is the prize. The prize is $5,000 for the first prize. That was a lot, especially when you were studying abroad. That was a lot of money. Okay, so what I did is, what we have to do is after you register, we have to do a lot of workshop. It's not like, okay, I just register and then I'll deal with it by myself. No, we have several workshops that you have to attend, all of them, to practice, to polish your presentation until the last day that you will be on the stage. So I have to attend all this workshop. And for this workshop, those workshops is just like torturing me. Because for each of them, we have to talk. We have to present. Not about my presentation, it's presenting something else because it's a training. So for all of the workshop, I just try to get through it and then try to learn a lot of it. So, there is a workshop that held in Taiwan that is called, that is held by Cai Yichen. Have you ever heard of it? Like, he was a group of people teaching a lot of students how to present, and the tuition fee, the price is so high. It's like 2万块钱台币每一堂课。然后and I thought that was like, no, that's nonsense. That was nonsense because they didn't give a very great lecture. But for my training, that was so. The training was very, very, how do I say that? It's very useful and it's more practical. So if later on, like in the future, if you guys want me to do it, I can try to copy all this training to here to NDMC for the juniors. Okay, so after all this training, I finished my presentation. So this is my presentation.
I am a liver transplant surgeon. It's always my greatest fulfillment when the patient restores their health and get back to the family. But sometimes, transplants are rejected and there's nothing we can do. This is because we don't completely understand how our immune system is regulated by our genetic code called DNA. 40 years after the first discovery of DNA, scientists finally decoded the human genome. The code comes out to be 4 fairly simple letters, A, T, C, and G. To build a human, you need 3 billion of them. As the most complicated creature, it's not surprising to have such a huge genome. But what surprises us is that only 1% is informative. So what about the other 99%? If they are not informative, then why do we carry such a huge amount of useless DNA? If we stretch out the entire genome, it's over 6 feet long. This is the height of an adult grown bear. How could this possibly fit into a single tiny cell? Actually, our DNA is extremely condensed and packaged into a substance called chromatin. The DNA is not squeezed, packed randomly like a bowl of messy spaghetti. Instead, it is folded into a very organized structure, territory by territory. Because our DNA is folded so tightly, not every part can be read. And my research focuses on how the structure of DNA changes in the immune response. In Batman, when Gotham City is in danger, they turn on the bat signal, and it's showtime. Similarly, our immune cells must turn on the danger signal in the form of changing their chromatin structure from closed to open to initiate the defense system. Let's do a small exercise. Can you see what's in my hand? Can you see the bat signal in my hand? Yes. So whether the chromatin is open or closed affects the activity of our immune cells. This is what we call the chromatin regulatory system. Amazingly, I have found the DNA regions, originally considered useless, change their chromatin structure, which allows our cells to fight off the deadly invaders. By sequencing the whole genome, I identify over 60,000 of these useful DNA regions, which could be the therapeutic targets to reshape our immunity. In fact, the 99% of human DNA, which were thought to be useless, carries enormous possibilities. Piece by piece, I firmly believe one day we will complete the treasure map of human genome, which will guide us to perfectly understand our immune system, so we can guarantee no rejection. Thank you.
It was a little bit embarrassing right now for me to rewatch my presentation, but I prepared it for 3 months, and that's what I did eventually, and I got the first prize surprisingly. And yeah, so that's my story. Like, okay, at the first time when I first on the stage in the US, I was blanked out, and I have terrible stage fright afterward. And after that, I decided to face that kind of problem that I have, and I joined this competition and finally nailed it. And this just like conquered my stage fright for a great deal. So that's why today I can talk about my life in UCLA in full, like the whole process. I can speak in English here very well. So the things that I want to tell you, okay, yeah, so I got the first prize at the year, and the next year I got the second prize for the UC campus-wide for the, compared to the other 10 UC, like UC Berkeley, UCSF, and UC Santa Cruz, blah, 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 for another 10 schools. Okay, so I'm here. I'm not here today telling you getting PhD is fantastic and you should do it. What I'm showing you today is how to explore your unlimited potential without fear.